thank you. Very nice presentation. This is not necessarily just for you. It's more for general question, and apologies that I haven't done my homework on polio. But has anybody looked, or is anybody aware of, was this something that was seen when polio was going around? Because clearly it was only a very small proportion of patients who had polio. And there must have been some variations across the seasons. But has anybody looked at that, or does anybody know?
Ellie, Amy is asking if you are able to repeat your question in reference to polio.
Oh, thanks, Amy. Sorry, I hope you can hear me. So my question was that is this kind of the variability of the polio paralysis, was that seen with polio as well? That maybe there was a season when there were less cases of paralysis reported, and still polio cases, poliovirus infections without polio.
Yeah. So if you look at the history of polio, there were many times when there were outbreaks of polio, but there was reduced paralysis. If I'm understanding your question.
Yeah, exactly.
Yeah.
So
and then there were many seasons, like, so for instance, as World War II ended and baby boomers came home and sanitation increased in the United States, there were many seasons where you enhanced the amount of paralysis that you saw. So like 1949 outbreak in Kansas City was one of the largest outbreaks in the United States that had ever reported.
And was there, Amy, any theory behind why this happened?
Well, so it was hypothesized that it's based on the sanitation conditions and the fact that the baby boomers had come home and there was a large population of people who had not been exposed to the virus. And we don't really have any samples anymore of the virus that was circulating at that time. It's not, they didn't really do a lot of these things, but they did do testing of the isolates that they did have using animals to look at serotypes and stuff. And some of it correlated with the fact that like type 2 doesn't cause paralysis as often as types 1 and 3, and you would have an outbreak of type 2 demonstrated by monkey tests and the protection, and then you would come back later, they would have another infection in that same area of a different serotype, and the amount of paralysis would be increased.
Well, this is a good opportunity to open the discussion and see how we are able to have a very good explanation for the 2022 paradox in the United States. So is any comment about that? Kevin, you are ready to raise your hand.
I'll ask a question stemming off Matt's question, that perhaps the virus hadn't changed as much in terms of number of mutations. So on the phylogenetic tree is more similar, but is it the number of mutations that matters or where those mutations occur? So like Amy, when we talk about reversion of OPV, do we know anything there? Is it about specific locations of mutations, or is it overall number? I would assume overall number equates to a higher probability of mutations happening in specific regions, but I'm just wondering if we need to study those specific changes that happened via mutagenesis studies or others to see what the significance of them are.
So for OPV, it depends. Again, it depends on the serotype. There's between. Well, nucleotide wise, there's probably 1% of or something or other. There's a lot of mutations nucleotide, but they don't affect amino acid changes. The one alteration that affects neuroinvasion properties of the virus is the same alteration within all 3 serotypes of the Sabin strains, which is at stem loop 5 of the iris. So it's 472C to U in type 3, 481AG in type 2, and I forget what type 1 is. But type 1 also has another alteration, which is in the VP1 protein. I think it's 525 and within VP1, and that is considered to be a secondary alteration. But if you look at the re-engineering of OPV into NOPV. So really there's the alterations that were introduced into NOPV2 and all of the NOPVs really have to do with reduced amounts of reduced frequency of recombination and dampening replication. So it's not the number of alterations, it's more critical where those alterations are. And this stem loop 5 alteration within the iris controls viral protein synthesis. So the hypothesis is that therefore you don't make a lot of protein, you don't make a lot of virions, therefore you have reduced viral fitness and you are allowing the innate immune response to clear the virus before it can gain traction in the body to be neuroinvasive. Did that make sense, Kevin?
Yeah. So it sounds like these regions, these critical regions, are really well known for polio and well studied and followed. How do we figure that out for EV68?
Do a lot of molecular genetics like my lab is doing and stuff. So not all entros is the same regions of the iris or the 5' untranslated region critical. And we don't really understand exactly the mechanisms of neurovirulence and neuroinvasion. So if you go to the GEPI website, there's an interview by Ananda at the Gates Foundation, and he describes the viruses that were recovered from patients that developed AFM following NOPV2 administration. And he calls them double recombinants and a reversion. And so if you think about that in terms of viral pathogen and viral biology, so they have they have 2 alterations within the polymerase, and they move the cis-acting replication element to the 5' untranslated region. And so obviously the first recombination event occurred at the 3' end of the virus to reintroduce a replication element, because otherwise the virus doesn't replicate. And then the second recombination event obviously remove the one at the 5' end. So until we really understand more about the molecular and at no point do I think like stem loop 5 that they changed, that fixation was removed. So until we fully understand the mechanisms of neuroinvasion, I think we're kind of hitting and missing, because obviously this recombinant with the stability of stem loop 5 has an alternative mechanism of being neuroinvasive.
Matt?
Yeah, well, and building off that, because I agree with all that stuff, is that this is part of why I was asking those questions of Dr. Mustafa about, like, were those whole genome sequences or not? Because I think, especially if people, as it sounds like many people on this very call are people who are building networks and increasing networks and trying to get this sort of data out of the humans, to be sure that we're not just doing like genotyping or serotyping or whatever kind of typing you want to call it. Really, it's molecular typing focused on those little stretches of VP1 that we often use to identify like which virus it is that we have in our hands. But, we have like these days relatively cheap sequencing platforms that can do the whole virus sequences. And this is only like a 7.5 KB stretch of sequence to do. And we do really need all of that stuff. Because Amy just told us, right? Like there's this part of the genome and then this part of the genome and this part of the genome. And when all 3 come together, you get, major attenuation or reversion of what had been major attenuation. And so we just to sort of encourage the people who are trying to think about this stuff critically and put these policies in place, I think it is important that we focus on those, because as much as I dabble around with antibodies and love to think about antibodies, and so of course I do love the surface proteins and all the mutations and interesting things that come from that. When we had our AFM working group seminar back in August or whatever that was, Carlos, I guess, when we had the 3 seminars and we had this session. If anybody was there for that or saw those recordings and saw, Bill Jackson's work from University of Maryland, he has really interesting and I think very compelling data that indicates, one of the ways that Enterovirus D68 might cause AFM is that one of the viral proteases might be cleaving the SNAP25 protein, which is basically the same thing that botulinum toxin does. And so it prevents your neurons from releasing, you know, the neurotransmitters that cause muscles to contract. So, you know, you could envision that a change in that viral protease, you know, amino acid, or one of those key things that it no longer can cleave the SNAP25, obviously you'll never see that if you're only sequencing the surface proteins. And so we'd never, you know, our dendrograms would look like we just said, they'd look like nothing ever changed. And we'd be sitting here wondering why we don't understand what changed and what's the difference. But that's just one thought around like why those, you know, why we need the whole genome. I think the other thing is that, you know, I definitely like to think in terms of what do we know for sure, and then what do we think we know? And I think we know for sure that we had a huge change in how the human population interacted for a couple of years. And that after that, we went from having a virus circulation period where we had paralysis and a virus circulation where we didn't have paralysis. And we are gathering data from people on this call who just presented that the virus didn't seem to change that much overall. So maybe the virus changed in key spots, or maybe it really is the hosts that changed and that's the key. And I mean, Sarah Kidd's guess was as good as my guess, which was like, you know, did the fact that everybody was immune naive for a while, their innate immunity systems were all calm, and then all of a sudden we got all the viruses at the same time, was that somehow actually protective? Because people are getting rapid fire viruses. I don't know. That's my favorite hand waving thing, and it's complete hand waving. Coming from a guy who was published with doctors Messakar and Grenfell, who are on this call, and Daniel Park's nice work, where we predicted there'd be a huge AFM outbreak, and we were dead wrong about that, right? So I don't know why anyone should listen to me after I just spoke and said all that stuff, but those are some thoughts.
Thank
Well,
you,
we
Matt.
do know that there is a ton, we do know how interferon susceptible some viruses are. And we do know from work by Marina Chumakov and a paper that Koisia published that was done in Iran, where they gave people OPV and it prevented infection with, for instance, flu. So like for his mother, her work has been published where she gave everybody OPV, and none of the patients who had OPV developed severe disease following influenza infection, if they even got influenza infection.
Sure. Yeah.
So we do know that when you have a bolus of viruses that are participating all at one time, and if you went and titered these people's blood, I'm sure that there's a ton of interferon in there. So I'm sure that you could be positive, but whether or not that was a robust, productive infection in terms of shedding, spreading, and the potential of being neuroinvasive is probably, you know, you could extrapolate that is significantly reduced by these papers that were done in the 60s.
And it's my favorite theory too, but the one thing that keeps me from feeling too comfortable about it, even though it is my favorite theory, so I'm agreeing with you. D68 was kind of the first wave of these back-to-back virus waves in a lot of places. It at least here, and I think in a couple other places in NVSN and such, again, please speak up, people who know the data. I think D68 was kind of like one of the first ones. So maybe like with that theory, then the RSV should have been not as bad, or the flu should have not been as bad.
Well, they were delayed.
I don't know. Yeah.
They were delayed, and I can make the argument that when 68 came up, Rhino had come up previously. Rhino was dampened, but not absolutely gone. I mean, it's not something that you can wear a mask and protect yourself against. It's more fomite transmission, which is why it used to be called nose picker's disease. And I can make the argument that Rhino was always circulating, and you still had high waves of SARS-CoV-2 infections. So I don't know.
So I have a question for Amy. What is your favorite explanation for the 2022 paradox in enterovirus and AFM?
So mine mine goes along with what Koistia's mother saw when she gave everybody OPV, is that we don't really understand how much interferon is required to dampen an infection, and that I would imagine that because SARS-CoV-2 was still circulating at a high degree, we were all constantly being vaccinated, and while the vaccine was quote unquote supposed to not trigger the innate immune response, I'm sure that there was some triggering to some degree, even if it was not detectable by your assay. And that is probably the majority of what controlled the infections from being neuroinvasive, is that it's so rare that they're neuroinvasive to begin with. And then when you reduce that another 100,000-fold, then you don't see anything above background.
Ellie, what is your favorite explanation for the 2022 paradox?
Yeah, no, I go for host as well. I agree with Amy, and I like her kind of a similarity that happened with polio and the hygiene theory, and immunology is not my strong side by any means. So I'm talking completely beside my field. But if you look at the studies, what Peter Openso, for example, has done in the UK with the human exposure studies with other viruses, it is the host response where they are when they get infected, which will determine the disease. So I think there's still a lot what we don't understand. So yeah, I go with that as well. I think it's the host.
Kevin, what is your favorite explanation?
I'm hoping it's the virus and it stays that way. But hoping, I don't think is the same as believing. I don't know what to think. I think preparing is better than predicting at this point, and we should all be braced for it coming back. And I think this group has done amazing work to recognize when outbreaks are happening. Unlike Matt, I don't feel bad about predicting or preparing for an outbreak in 2022, even though we didn't see AFM. I think EV68 in and of itself from a respiratory standpoint creates huge morbidity that goes completely under recognized. I really congratulate Hiba for the work at Hopkins. I think that figure you showed, which is not just the rhinoenteros positive on RPP, but all of our patients who are undergoing these fluid assays that don't even pick up rhinoenteros, you showed a similar percentage positive for EV68. And I really think the real mover in this field from development of therapies and vaccines is going to be when people recognize the respiratory burden of this virus. And I really think it's going to take inclusion of an EV68 target on one of the commercial platforms that allows us to really recognize this is a player just like, you know, flu, RSV, and HMPV, the big respiratory viruses in children. And I think once we start to realize what a huge respiratory burden it has, it'll really help us from an AFM standpoint to get more resources towards combating the kind of overall pyramid of morbidity that it causes. So that was a hedge, but I really hope I think the one thing Amy and everyone else will tell you is that it is not beneficial to EV68 to cause neurologic disease. It's a bystander effect. A virus doesn't transmit from the CNS onward. So I would love it if mutations have happened that made it less neurovirulent. Even if it continued to cause respiratory disease, it would be a very welcome absence of seeing these devastating cases.
Kim, what is your favorite explanation for the paradox?
I agree also with what Amy said, and I think we also need to look at what the host is doing. We are only looking now at the samples, the host samples, but I think maybe we should go forward to, when we have these cases, to also collect to try to characterize the immunology and the sequencing of the host and see where it's why it's causing AFM in one case and not in the other case. So I think it's a host that plays a major role in this as well.
Peter, what is your favorite one?
Yeah, putting me on the spot here. I was just enjoying listening to it, really. I'd go for aseptes. But I think the difficulty is that it may be quite difficult to predict from the protein sequencing.
Eva, what is your favorite?
I
I think if we assume that the paralysis is due to neurovirulence of the virus, if we actually believe this hypothesis, then I think prior exposures and pre-existing immune responses are contributing a lot to the presentation of the disease in specific age groups and in specific years. And this is actually the hypothesis we would like to test. And I actually see that Matt is also in the call. So this is actually a proposal that we worked together on. And it will be really interesting to look back and see based on our cohort from the previous many years and based on the ages of the patients, what type of the pre-existing antibody repertoire, among others, that correlated with a specific outcome for infections with the virus. Then I agree that there should be also like a large host component that makes some more predisposed to the neuronal kind of version of the infection, because at the end of the day, the percentage of those who will have paralysis is not that high. So it's a complex scenario, but I really do believe that we should look at the contribution of pre-existing immune responses due to exposures to all different types of enteroviruses, rhinoviruses, and others.
Great. Sarah, what is your favorite explanation?
I knew it was coming. I don't think I can expand on what's already been said. I think I outlined what the three just options at at the beginning, but I think I agree with what Amy, and Amy has a much more eloquent and logical reason for it. It is able to explain that a little bit better. But yeah, I think it's something having to do with load of infections during the summer in 2022.
So Brian and Daniel, the team modeling these situations, what, after listening all of the discussion, what do you think? What is the best explanation for the paradox?
What a great discussion. I mean, it's great to hear Amy's wisdom. Actually, I think that's very interesting. I hope it's the virus, and the interferon idea is a very interesting one. I'd still got this nagging feeling that I'd like to understand. It's a very transmissible virus, this, and the age of primary infection is probably very young, and the age of AFM was a bit older, and I'd like to understand that age matching and what the age structure of cases and of primary and secondary cases was in 2022. There must have been lots of first infections. So I'm not saying that is an explanation as a host factor, but I'd like to understand that a bit better. But I certainly hope it's the virus.
Is anybody else like to give an opinion about explanation of paradox?
How about you, Carlos?
I think that is the host and the immune response. And unfortunately, when you are dealing with infectious disorders that affect the central nervous system, you are dealing with three factors: is the host, obviously the genetic background, the virus, and the immune response. And unfortunately, sometimes try to separate those is a little bit challenging, but I believe that we need to pay attention to the host. And I think that polio is a fascinating situation that probably we need to go back and take a look in details, all of those papers that were published before. But I think that what we experienced with COVID-19 infection and later other significant infection actually is that influencing the immunological response to viruses. And I think that is a little bit complex, but I believe it's the host, the genetic response, the immunological response, and as well as the virus. It's going to determine the immunological response. All right, it's 3:21. Thank you so much. It was a very interesting discussion. Thank you, everybody, and thank you to our colleagues in Europe that are staying late tonight for participating in the discussion. And we will send you an invitation for the next meeting. We are open-minded about the next meeting, particularly because we are just expecting to see what happens in the next few weeks. If we see enterovirus circulating, and if we need to be alert. And so we will be in communication, and we will alert you